Hi, this is our Cell Theory and Life 101 Day 3 set of notes. It'll be kind of a shorter time, and then in class today, I had students do some independent work. So you'll get to do that too. Today we also did some water quality testing, so everybody got out their experimental design diagram packet for fertilizer. It's the little packet that has the jars, the pictures of the jars. And so we remembered that our question that we're trying to answer with the experiment is, how does nitrogen that is found in fertilizer impact water quality and plant growth? And remember, this is because we have been studying the nitrogen cycle, and we want to understand, in some ways, how do humans have an impact on the nitrogen cycle? All right, so the only thing that was different in each jar, A versus B versus C, was the amount of fertilizer that we put in. And I asked groups to let me know what kind of differences they saw between jar A, B, and C from the very first day of testing. And even though that was early on in the experiment and the jars hadn't been sitting for very long with their Alodia, a lot of people said that jar C already had lower dissolved oxygen levels. All right, so I asked everybody whether they liked pancakes or waffles, and it was kind of mix and match, but that was just our get warmed up and talking question. So, even though it says we're going to practice using the microscope and practice scientific drawing skills and then also measure and record water quality, we really did not get to do any more microscope work on this day. Those were my hopes and dreams, and it didn't quite happen. So just pretend like I didn't have that in the beginning of my what. So instead, we really did just measure and record water quality, and we practiced our cell theory stuff. Okay, it is important that we know how to use a microscope so that we can accurately observe cells, so that we can understand the parts of cells and how they work together. All right, so just a reminder, if you were not in the classroom, you will want to get data for week 2 from your team members. So in class, we wrote down either February 14 or February 15. And we switched our jobs. So these were the jobs for this day. And if you need practice measuring nitrates, you could come during Falcon Focus. But it will rotate around, and 3 people are always measuring nitrates on any testing day. All right. Then just a reminder, we test for temperature, dissolved oxygen, pH, nitrates, and clarity. And we make some qualitative observations about how do the plants look inside there. Okay. The recorder is the person who's going to bring the packet over to the station, and that person will be recording data for all of the jars. That person is also going to check water clarity, because we do have some differences in water clarity. And then, of course, once you all get back to your desks, you're going to share your data. And anytime we're using chemicals at all, or working with water and water quality testing, we're going to be wearing goggles. All right, so I gave everybody 12 minutes to do the water quality testing. We're not going to do that here. And then once everybody was seated and settled, we talked a little bit again about what is cell theory. So that's going to be a big component of our upcoming test. So what is cell theory? It's the theory that all living things are composed of one or more cells. The cell is the fundamental unit of life. And here's what it says: that which originated from pre-existing cells. What that means is that all cells come from other living cells. They don't just spontaneously appear. All right, so a reminder, after you did your independent work during our last class, you watched a video about the wacky history of cell theory. And so hopefully you remember the 3 parts. Try to think of them yourself right now. Click pause on the video and then come back and see if you were right. So for cell theory, all living things are made of one or more cells. The cell is the most basic unit of life, and things that are smaller, that are part of cells, are not considered to be alive. Okay? And then number 3, all cells come from other cells. So you wrote down some of this information in your lab book the other day after you were doing independent slides. So we're going to check your work right here. And in 1665, we have Robert Hooke, and he was the first to see cork cells. Sorry, you can't quite see that. He was the first to see cork cells. So he saw those through his microscope. They were not alive. He was looking at cork, which is basically dead bark. And he just described what he saw, and they looked like the cells that the monks lived in the monastery. But you can think of dorm rooms, you could even think of prison cells, but kind of uniform shaped open spaces. And then in 1673, so only a couple years later, you can see, Anton van Leeuwenhoek observed living cells through a microscope. He's the one who scraped his teeth, and he got bacteria from his teeth, and he was looking at those living cells through the microscope. All right, then please double check your timeline, because in 1838, Matthias Schleiden, discovered that plants are made of cells. And in 1839, just a couple months later, Theodor Schwann discovers that animals are made of cells. They are super close together in time, but I'm going to say Schleiden came first with his plants are made of cells ideas. So please make sure that's what it says on your paperwork as well. I think something was not perfect on our slides last time. I think something was switched. So also it depends on what research you read. But we're going to say Schleiden came first, and he knows that plants are made of cells, and Schwann came second, and he knows that animals are made of cells. Okay, he looked at water, and he actually called them animalcules. So 1855, also not too terribly far away from Schleiden and Schwann, we have Rudolf Virchow, and he discovered that all cells come from other living cells. So if you remember from the video, when, let's see, no, I don't remember which one. When one of our guys was looking, he was convinced, and I think it was Schwann, he was convinced that the cells just spontaneously appeared on his slides. He was convinced that they just appear, they spontaneously generate. But Virchow believed very strongly that that's not the case. All cells come from other cells. And that's because he was looking at live cells under his microscope, and he saw some cells divide. He actually saw them divide and go from 1 to 2. All right, so you'll notice that these are actually, in the timeline, they are alphabetical by last name. So Hooke, Leeuwenhoek, Schleiden, Schwann, and Virchow. It's just helpful to remember that they're alphabetical. Also, I'm going to give you some ways to remember which scientist came up with what. It didn't happen overnight. You can see that Hooke and Leeuwenhoek are pretty close in time. They're in the 1600s there. And they actually corresponded and had some communication. And that'll come back when we look at nature of science. The nature of science is that it is a complex social activity. So we do have scientists working together here. Then, if you notice, it's like 150-ish years later, we end up with Schleiden and Schwann and Virchow. They're kind of grouped together in time also. So there have been advancements in the ability of compound microscopes to focus, and they are able to see different things. All right, so back to nature of science. We did this at the beginning of the year, and we have these basic ideas about the nature of science. We believe that the natural world can be understood through exploration and experimentation, right? But we need evidence to prove our ideas. Science is a blend of logic and imagination, and so some of those guys making the compound microscopes, they had to be kind of imaginative and creative and put lenses together to have a better magnification level. Okay, scientific knowledge is durable. We are still using a lot of that cell theory now in our time in the 2000s, and so it has lasted over time. But it's also subject to change. So if one scientist believed that cells just spontaneously appear, later on, Virchow was able to do some observations, and he realized that that is not true. It's not that they just appear, they are dividing, and when the cells divide, we go from 1 to 2. Okay, science is a complex social activity, and we talked about that and how scientists sometimes work together. Sometimes they actually argue a little. but they are at least aware of each other's ideas most of the time. All right, so I've got some mnemonics that might help you remember which scientist did which thing. So Schwann sounds a lot like swan. That's an animal. That's the guy who discovered that all animals are made out of cells. All right, get ready for this one. It's a stretch. Schleiden. Pretend that he's sliding through the grass. He's sliding through the grass on like a fake sled there. And grass is a plant. He's the guy who realizes that all plants are made of cells. And we have Van Leeuwenhoek. And remember, I say Leeuwenhoek. A lot of people will say Leeuwenhoek. Some people will say Leeuwenhoek, and some people will say Leeuwenhoek. It depends. I'm going to say Leeuwenhoek almost every time. So L for Leeuwenhoek is the first guy to see living cells. And then we have Virchow. It's a little bit like Virchow. And so he's chopping something into two pieces. So he realizes that cells come from other cells because he sees one cell divide into two different pieces or two different cells. So that might help you remember which one's which. And then back to Hooke. Just remember, Hooke looked through that microscope. He was looking at cork. He didn't really know the parts that he was looking at. All he did was he just named them. He said, hey, that looks like a whole bunch of cells, like where the monks live. And not too long after that, we have Leeuwenhoek looking at live bacteria. Okay, so Hooke discovered cells because he was looking at cork. And, cork is actually just basically dead bark from trees. All right, I let people look for a mouse on this. You can pause if you want to. And then in your blue lab notebook, don't forget you're bringing that back and forth every day. In your blue lab notebook, we are on lesson 2 right now, Life 101 right there. We're investigating cells as systems for life processes. So remember that we've studied ecosystems, which are very large, right? And now we're going to shift our focus to things that are quite small. So small, in fact, that you must use a microscope and a wide variety of models to investigate them. So to help make the shift in scale, we're going to arrange some things in order we actually arranged in the classroom from smallest to largest. So I gave everyone cards that have these words or phrases: organ system, atom, biome, population, organ, cell, ecosystem, biosphere, cell. Maybe it's saying cell, organelle. I just wrote organelle on my cards. Organism, tissue, molecule, and community. So I gave out cards and let people sort them on their desks. So I gave only 3 minutes for that. And then we went through and figured out what was the order. So from all of those words, the smallest is going to be the atom. Okay, and then the next size up is a molecule. Notice a molecule is several atoms bound together, not just one atom, right? So molecule's a little bigger than atom. And then we have organelles. Organelles are specialized little parts within a cell that have a specific job. So chloroplasts are inside the cell, and they have the specific job. That's the location where we're going to do photosynthesis. So chloroplasts are inside a inside a cell, which means that they're smaller than a cell, right? And then here we go. We've got our cell. A cell is living. This is the smallest living thing that we know, okay? There can be live things that are exactly one cell big, or we can have multicellular organisms as well. All of these smaller things are not alive. They are smaller things, but they are not alive, okay? So they're not cells. Here's our cell. Next up, we have tissue. So that means that you've got a lot of cells that are doing the same thing. So you might have stomach lining that is tissue, or you have skin tissue, or you have muscle tissue, right? So you have a whole bunch of muscles that are doing basically the same thing. And then those things might be all the same kind of tissue within an organ. So we have different organs like our pancreas and our liver and our lungs and our stomach. And once you have a bunch of organs working together, you're going to end up with an organ system. So that's like the digestive system, right? That uses the stomach and the intestines and different organs so that it can accomplish a job. And the digestive system is accomplishing digestion, right? All right, after that, our next biggest thing is an organism. So an organism is made up of all of the smaller things that we've talked about. Okay, when we have a whole bunch of the same organism living together, that's a population. And then when we have lots of different types of populations, we're going to say that that is a community, the different species that are living together. Where are they living? They're going to be living in an ecosystem. So in the ecosystem, we are talking about both the biotic and abiotic things. And then the next size up, we're going to talk about biomes, which we have talked about in our class. And finally, the biggest one of those categories was the biosphere. So we've got everything organized by size. And when we've been looking at big things, ecosystems, biomes, biospheres, now we're going to start looking at the small things, okay? And in fact, you can see investigating cells as systems for life processes. We're going to be investigating cells for sure. And here again, this is a listing of all of those different cards that we sorted in the classroom in their order from smallest all the way to largest. All right. We're going to study the cell, of course. So here's a video. Let me see if I can turn myself off. Hold on.